Might be safe at home? Today in this online session, I am again back with part 4 of molecular basis of inheritance of class 12. The topics for today's online session are the genetic material is DNA, experimental proof for that, properties of DNA, material, DNA versus RNA and RNA world. So children, let us start with first topic of today's session, that is the genetic material is DNA, and for that experimental proof. The genetic material is DNA is proved by experiment done by Alfred Hershey and Martha Chase in the year 1952. Experimental material taken were bacteriophage and bacterial cell. These bacteriophage are those viruses that infect bacteria. When the bacteriophage attaches the bacterial cell, then its genetic material enter into the bacterial cell. And as soon as this happens, bacterial cell treat the viral genetic material as its own and thus produces more virus particles. Hershey and Chase worked to discover that out of protein or DNA, what has entered the bacterial cell. To prove this, they grew some viruses on a medium that contained radioactive phosphorus and some viruses on a medium that contained radioactive sulfur. It was found that viruses grown in the presence of radioactive phosphorus contained radioactive DNA but not radioactive protein, because DNA contains phosphorus but protein does not contain phosphorus. Likewise, viruses grown on radioactive sulfur contain radioactive protein but not radioactive DNA, because DNA does not contain sulfur. Both these radioactive bacteriophages were then allowed to attach to E. coli bacteria in separate experimental setup. As the infection proceeded, the viral coats were removed from the bacteria by agitating them in a blender, and virus particles were separated from the bacteria by spinning them in a centrifugal. It was found that bacteria which was infected with viruses that had radioactive DNA were radioactive. This indicates that DNA was the material that enters into the bacterial cell from virus. And bacteria that were infected with viruses that had radioactive proteins were not radioactive. This indicates that protein did not enter the bacteria from the virus. Thus, DNA is the genetic material which enters the bacteria from the virus. The given figure shows about the Hershey and Chase experiment. Here the three steps can be clearly seen, that is infection, blending and centrifugation. When bacteriophage is grown in radioactive sulfur, then no radioactive sulfur was detected in the bacterial cell, but radioactive sulfur was detected in the supernatant. And bacteriophage is grown in radioactive phosphorus, then radioactive phosphorus was detected in the bacterial cell and no radioactive phosphorus was detected in the supernatant. DNA being heavier than protein. Now It is about properties of genetic material, that is DNA versus RNA. Genetic material should be able to replicate and should be chemically and structurally stable. Genetic material should provide the scope for mutation and which is necessary for evolution. Genetic material should be able to express itself in the form of Mendelian characters that passes from parent to offspring. As far as both the genetic materials DNA and RNA are concerned, DNA is chemically less reactive and structurally more stable as compared to RNA. Thus, DNA is a better genetic material than RNA. Also, DNA is preferred for a storage of genetic material and as compared to RNA. But for transmission of genetic information, RNA is better. RNA being unstable, it mutates at a faster rate. Viruses having RNA as genetic material and having shorter lifespan mutate and evolve faster than DNA. RNA can directly code for the synthesis of protein, hence can easily express the characters. For synthesis of protein, DNA is dependent on RNA as you have seen in central dogma. Thus, the protein synthesizing machinery has evolved around RNA. These are the figures showing differences between RNA and DNA. You may find the differences in the type of sugar, type of bases present, and single stranded in RNA and double stranded in DNA. This is also about the DNA versus RNA. Now, the last topic of this online session is RNA world. RNA was the first genetic material. It acts as a genetic material as well as a catalyst. Essential processes like metabolism, translation, splicing, etc. are evolved around RNA. RNA acts as catalyst, so reactive as well as unstable. Therefore, DNA has evolved from RNA. So children, this is all about today's session. Thank you. And for any kind of doubts, please write your question in comment section.